Good morning, students. Myself, Dr. Balasubramaniam, working as assistant professor in the Department of Zoology, Government First Grade College in the PG Center, Chintamani. So today we are going to discuss about the reproductive system in Unio. Now this topic is belonging to the second semester BSc Zoology syllabus and in the type study of Unio. So here we conveniently divided this topic into two sessions. One session being study of the reproductive system of Unio, the second being the life history of Unio. So in this session, we are going to study about the reproductive system of Unio. Now, reproductive system, in the case of the Unio, it is dioecious. Unio is dioecious. So Unio, or the freshwater mussel, is a dioecious animal. That is, the sexes are separate. But there is no sexual dimorphism. Sexual dimorphism is absent. Now, externally, we cannot distinguish between the male and the female. So that is the meaning of sexual dimorphism. Sexes are separate, and Unio is a dioecious animal. Next point: the gonads are testes in the males and ovaries in the case of the females. Now, the gonads are paired, large, highly branched structures lying in and around the intestinal calyx in the visceral mass above the foot. Now, in the visceral mass, we find the digestive system as well as the kidney or the metanephridia as well as the gonads, and also we observe the coelomic cavity and we observe the digestive gland. Now, in these mollusks, so the body has a main thing, that is the viscera. Now in the viscera we find all the important organs like, for example, digestive system and excretory system, reproductive system, and even the nervous system as well. But the respiratory system is found outside the organism, and outside the visceral mass. It is found in between the mantle. Now, in between the mantle, we find the respiratory structure that is called tenidia, or the gills in the case of the Unio. Now, therefore, this visceral mass has the lot of important organs as far as the Unio is concerned. Next point: during breeding season or reproductive season or mating season, the gonads become greatly enlarged. The gonads, that is the reproductive organs or the sex organs, becomes greatly enlarged and conspicuous. The testes being brightly whitish in color, while the ovaries are reddish in color. So there is a color differentiation in the case of testes and ovaries. Then the coelomic lining of the gonads proliferates to give rise to spermatozoa in the males and eggs in the females. As far as we know that any gonad has the primordial germ cells or the sperm mother cells or spermatogonial cells. These spermatogonial cells or the sperm mother cells or the primordial germ cells, they undergo mitosis as well as meiosis to give rise to the sperms. Similarly, in the ovary also we find the primordial germ cells or the oogonial mother cells. So they undergo the process of mitosis and meiosis. Now that three phases are there. Now one is the phase of multiplication, phase of maturation, and first is the phase of multiplication, phase of the growth, and phase of another phase, I can't remember that one. So here three phases are there. Then this spermatogonia in the males and females. In the males we find that is the three steps are there. Next, each gonads has a short duct called vas deferens in the males and oviduct in the females, and these males, which has the vas deferens, and females has the oviduct, they opens into the suprabranchial chamber of the mantle cavity near the excretory pore. So that means the gonads opens outside in the males by vas deferens and oviduct in the case of females. So that is into the suprabranchial chamber, and this open to the mantle cavity near the excretory pore. Next character are this explanation. So there is the accessory reproductive structures are not found in the case of the Unio. Now, next here, so I just recollect now. 3 phases are there in the what you call this one, the sperm production. One is the phase of multiplication, phase of growth, and third is the phase of maturation. So 3 steps are there, either in this protogenesis or oogenesis. 3 stages are there. First is the phase of multiplication, phase of growth, and phase of maturation. So here, continue with the topic. Now each duct open, whether it is oviduct or sperm duct, opens into the suprabranchial chamber of the inner gill lamina of its own side. Now this opening is called genital aperture, situated just in front of the renal aperture of the ureter. Now there are also no copulatory structures as in the case of the Unio. That is, like in the case of the higher animals, where there is an intermediate organ for the exchange of the gametes. Now in this Unio, you don't find the copulatory organs. So that is the meaning. There is no copulatory organs, as well as there is no accessory organs. Only there are gonads are there. As in the case of the higher animals, we don't find this copulatory structures and accessory reproductive structures. Only we find the testis and the sperms, testis and ovary, and these ducts are there attached to the gonads. In the males, the sperms are shed through the genital aperture into the suprabranchial chamber and expelled from the body along with the outgoing currents through the excellent siphon. So that is the meaning. So here, the males, the sperms are shed through the genital aperture into the suprabranchial chamber and expelled from the body along with the outgoing water currents through the excellent siphon. So in the Unio, we know there is inhalent siphon and exhalent siphon. Now inhalent siphon is slightly larger than the exhalent siphon. So inhalent siphon is meant for the incurrent of water, exhalent current is meant for the exit of water. In addition to the respiration, feeding, and the gametes are also expelled through the waters, through the exhalent siphon. Now next character are the next points. Some of the sperms enter through the inhalent siphon of the female purely by chance. So these sperms enter the inhalent siphon of the female Unio purely by matter of chance. Further, the eggs shed through the genital aperture into the suprabranchial chamber do not leave the body but are carried to the water tubes of the outer gill lamina. So here, as the male also release the sperms, the females also release the eggs shed through the oviduct, that is the genital aperture, into the suprabranchial chamber. But here, the sperms come outside in the Unio, but the eggs doesn't come out from the body of the Unio. So they stay in the suprabranchial chambers and they carry the water tubes into the outer gill lamina. Now here, the eggs are deposited in the water tubes of the outer gill lamina. That is the meaning. Next, development of the fertilized eggs or zygotes proceeds within the water tubes of the gill lamina, which are greatly enlarged due to the accumulation of large number of embryos. So here, these water tubes of the gill lamina provide a place for the early development. Now, in the case of the Unio, we find the water tubes in the gill lamina where the eggs are deposited or eggs are placed. Now the sperms enter through the inhalent siphon and they fuse with the eggs to form the zygote. Thus, the water tubes of the outer gill lamina serve as the brood pouches or marsupia. So here the gill lamina serve as the brood pouches or marsupia in the case of the Unio. Now therefore, the females are easily recognized at this stage by swollen appearance of their outer gill lamina. Now here, this is a diagrammatic representation of the Unio, a Liquidium larvae, in the development of the Unio, where you find the hinge and single adductor muscles and two shell valves. So there is a bivalvular pelecypoda, and there is a mantle and sensory bristles and a hook for attachment to the fish, this ectoparasite. So this Liquidium larvae act like the ectoparasite attached to the fish by the means of hooks, and byssus thread is also found for the purpose of the nourishment. So summary in this topic, so here we are going to discuss what this Unio is dioecious, sexes are separate, and there is no sexual dimorphism. Development of fertilized egg or zygote proceeds within the water tubes of the gill lamina, which are greatly enlarged due to the accumulation of large number of embryoes. So in this session, we saw about the reproductive system of Unio and how these gametes are released, particularly with reference to sperms, whereas this eggs doesn't come out from the Unio body, and these sperms, by matter of chance, they enter the inner siphon, and now they mate with the egg, which is found in the water tubes of the gill lamina, where there is a fertilization and the zygote is formed. Now in this chapter or in session, we saw these many topics. Next session, we see about the further aspects of the life history of Unio. Thank you for listening. And here, last slide, we have the references. See here. See, R.L. Kodpal, that's the invertebrate textbook, Jordan Verma, Yakamana Tayar, and Dame and the Bendesan Prasad. So these are the textbooks. And of course, so I took the information from the net, there is a Google, these pictures, photos particularly. But information I took from the Kodpal textbook. Thank you for listening.